Welcome back to Chapter 6. In this last section, we're going to discuss viroids, virusoids, and prions. These are unusual infectious agents. They're not viruses, but they can still cause disease. And by the end of this section, you'll be able to describe the unique characteristics of viroids, virusoids, and prions, and how they differ from viruses. Viroids were discovered in 1971 by Theodore or Ted Diner, and he was a plant biologist from Switzerland who actually passed away recently in 2023 at the age of 102. Viroids are the smallest known infectious agents. Unlike viruses, viroids have short, circular, single-stranded RNA only. There is no protein coat, no capsid, so unlike viruses, viroids do not have a protein coat to protect their genetic information. They replicate using the host cell machinery, and they cause a variety of different plant diseases, such as potato spindle tuber, tomato plant disease, avocado plant disease, and peach disease, peach latent mosaic disease, and this can actually also affect plums, nectarines, apricots, and cherries. Viroids spread by contaminated tools, seeds, or insects, and they can cause major agricultural losses. So this is a picture of potatoes that have been affected by that potato spindle tuber viroid, and you can see that the viroid actually takes over the host machinery and stunts and prevents normal metabolism of the plant cells. This will interfere with the pathways that are usually critical for normal growth. And the viroid can be spread when infected knives are used to cut healthy potatoes. And I wanted to show you how small the genome is for these single-stranded RNA molecules. And they're shown over here. These are single-stranded circular RNA molecules that have kind of collapsed on themselves. And they're tiny compared to this larger circular molecule, which is DNA from a bacteriophage. Remember, bacteriophage are viruses that infect bacteria. So for our viroid checkpoint, remember the genome is a short, circular strand of single-stranded RNA. And in that previous picture, they kind of looked linear instead of circular because these tiny circles, like my hairband, can collapse upon itself, making it look like a line when it's actually a circular form. Virusoids are similar but have an important difference. They are also small single-stranded RNA molecules, but they cannot replicate on their own. They require a helper virus in order to replicate. In plants, virusoids use sobemoviruses as their helper. So virusoids are different from viroids because virusoids are packaged inside a helper virus and require their help in order to cause disease. In humans, the best example of a virusoid is the hepatitis delta virusoid, or HDV. HDV is a circular RNA virusoid that requires the hepatitis B virus as its helper. And when patients are infected with both, co-infected with both, so infected with both the hepatitis B virus and that hepatitis delta virusoid that's packaged within the hep B virus, it causes a more severe liver disease. So let's compare viroids and virusoids. And first, their similarities. They're both subviral agents, so they're both smaller than viruses. They're both made of small, circular, single-stranded RNA. They do not encode proteins. There's no capsid. They don't have enzymes. They're just RNA. And replication depends on that host cell machinery. And they can also both cause diseases in plants, although virusoids differ because they can also cause disease in humans, such as that HDV virusoid. The main difference between the two are that viroids are self-replicating RNA. They do not need a helper virus, whereas in virusoids, they cannot self-replicate. They require a helper virus to replicate. Prions are our last type of acellular pathogen, and they are completely different. They are infectious proteins. There is no DNA and no RNA in their structure. They were discovered by Stanley Prusiner in 1982, which later earned him the Nobel Prize in Physiology and Medicine. and I'm not sure if he still is, but he was the director of the Institute for Neurodegenerative Diseases research lab at UCSF here in the Bay Area. So it turns out that we have normal protein in our brain called PRPC. This is, C stands for cellular, so cellular prion protein. But if somehow the misfolded form, PRPSC, gets into your blood and gets into your brain, it can convert the normal protein into the PRPSC misfolded version. And this stands for, SE refers to scrapie, because it was named after scrapie in sheep, where it was first discovered. This leads to plaques in the brain tissue, causing degeneration of the brain tissue. And diseases caused by prions are called transmissible spongiform encephalopathies, or TSEs. In humans, these include Creutzfeldt-Jakob disease, fatal familial insomnia, Kuru, and GSS, and I'll show you a table in a bit. In animals, this can lead to mad cow disease, scrapie, or chronic wasting disease. Unfortunately, there is no cure, so this is always fatal. And what's even worse is that prions are extremely difficult to get rid of, to destroy. They are resistant to heat, chemicals, and radiation. Almost nothing can destroy a protein that's already misfolded, except for complete incineration. And here I've written out what they stand for again. Prion protein cellular form is that normal form, and the scrapie form is named because they first discovered scrapie or prion protein in sheep, which causes scrapie. Here we can see how the interaction between PRPSC and the normal prion protein can cause the normal endogenous, so normally found PRPC, to convert into the bad form, ultimately causing brain degeneration. And you can see all of these plaques, right? These holes in the brain. Here's a picture from our book comparing the normal brain with a brain from a patient who has Creutzfeldt-Jakob disease, right? This is a fatal disease caused by the prions. And then over here we have tissue, and we have normal brain tissue compared with a patient with CJD. And you can see those sponge-like lesions, those holes in the brain that make it look like a sponge. Transmission of TSEs, transmissible spongiform encephalopathies, unfortunately can arise in several ways. So different ways prion diseases can arise, or prion diseases can arise, is sporadic. This means there's a random mutation of that cellular prion protein to form the scrapie form. Familial is inherited, when you inherit the gene mutation. Variant Creutzfeldt-Jakob disease occurs when you eat contaminated beef. So cows might be infected, and if you eat the meat from that cow, you can get sick. And unfortunately, fully cooking the beef does not work because, remember, prions are heat resistant. This is different from Creutzfeldt-Jakob disease when we add the V. So V, the variant form, is from eating the beef. Iatrogenic, this is an I, not an L, is through contaminated surgical instruments or grafts or transfusions from patients who were infected. And Kuru is through historically spread through ritual cannibalism, which is when humans eat other humans. Unfortunately, as I mentioned earlier, prions are extremely hard to destroy. Normal sterilization methods do not work, and the current gold standard is complete incineration, so burning something completely to ash of the contaminated materials. Although there are developments, so they're trying to make this enzyme called prionzyme that destroys specifically the PRPSC form, but again, the current gold standard is still complete incineration. And our book has a table summarizing the different TSEs, the transmissible spongiform encephalopathies. You'll see CJD next to the terms I mentioned earlier. CJD is named after the two physicians who first described the disease in the 1920s. Creutzfeldt and Jakob were separate German neurologists. They were both actually independently studying this rare, rapid, progressive neurodegenerative disease that had symptoms and signs like dementia, movement problems, and sponge-like changes in the brain. They didn't know until later that they were actually both studying the same disease, so it was named after the both of them, Creutzfeldt-Jakob. For our prion checkpoint, remember prions do not have a genome. They are just infectious proteins. There's no DNA, no RNA. In patients, prion diseases cause dementia, loss of coordination, and brain degeneration. Unfortunately, prion diseases are untreatable and always fatal. So public health measures really focus on prevention, controlling infected meat products, regulating medical procedures, and enforcing strict sterilization protocols or incineration when necessary. Importantly, and luckily, there's no evidence for spread through casual contact. All right, let's recap these viroids, virusoids, and prions. For viroids, they are the smallest known infectious agents. They consist of only short, circular, single-stranded RNA. There's no protein coat, no capsid. They are self-replicating using host machinery, and they can cause plant diseases like potato spindle tuber or avocado disease. For virusoids, these are also small, non-self-replicating single-stranded RNA, so they cannot self-replicate. Remember, they need a helper virus, like the sabinoviruses in plants and hepatitis B virus for HDV in humans. They are packaged inside the capsid of the helper virus, and they can be found in plants, but also in humans through that hepatitis delta virusoid. For the prions are infectious proteins, right? There's no DNA, no RNA. We have that misfolded form, the PRPSC, which converts the normal PRPC cellular form into that rogue form. They can cause transmissible spongiform encephalopathies, those TSEs, in animals and humans. And unfortunately, prions are resistant to heat, chemicals, radiation. They're super hard, very difficult to destroy. And it always causes a progressive and fatal disease. There is no cure. And I wanted to take a moment to compare bacteria, which are cellular, to all of these acellular pathogens. For their genome, remember we have double-stranded DNA in bacteria, whereas in viruses they can be either, and they can be in their single-stranded or double-stranded form. Viroids had single-stranded circular RNA, so did virusoids. Prions have no DNA or RNA. For protein capsid, there's none in bacteria. There are in viruses, but none in the prions, viroids, or virusoids, although virusoids are packaged inside the capsid of the helper virus. For replication, this is independent through binary fission for bacteria. These require host cells. The virusoid requires that helper virus and the host cell. And how prions kind of replicate, they don't really replicate, but they convert normal cellular prion protein to that scrapey form. Notice none of these acellular pathogens have their own metabolism. We talked about a variety of diseases that can occur. And for treatment, we can take antibiotics if the bacterium is not resistant. For viruses, we have some antiviral treatments, but we can also have vaccines for preventative care. For viroids, this is really through agricultural control, such as preventing the spread of contaminated seeds. Here we can try to target the helper virus. For example, the hepatitis B vaccine, that virus vaccine, can help prevent the virusoid infection. And then unfortunately for prions, there's no cure. We can prevent by incinerating things that can be contaminated. All right, and that takes us to the end of Chapter 6. In Chapter 7, we're going to look at some organic macromolecules. Thank you for watching.